to talk about precision-cut liver slice. And as Abby mentioned, I hope this will be a informal platform to exchange ideas. I cannot say I'm the expert of PCLS. I just started using PCLS about 2 years ago, and I would like to share what I have learned, and I would like to hear what you have done. I know that most of you do not have your video on. For any of you who have worked on precision-cut liver slice and are actively doing this, can you just put in the chat box and tell me that you're doing this? And I would like to hear your thoughts of your experience as well. All right, so we'll get started. So this is my disclosure. So for a lot of you probably know this. This is probably not news to most of you. Unfortunately, the life expectancy in the US has been falling behind a lot of the major developed countries. And for about 10 years till COVID, US actually has experienced about a decade of decline in life expectancy. Why I'm talking about this? One of the contributors to the reduction in life expectancy is because of liver disease. So this is a study done by my friend and a colleague, Elliot Tepper at the University of Michigan, and he summarized the life expectancy and liver disease related mortality. And you can see that in the figure on the left, you can see the mortality cross age group as well as gender. There is an increase of mortality from liver disease in almost all age group, especially the people who are in the 25 to 34 age bracket, and especially women has a higher than men mortality there. And this is Massachusetts. Massachusetts is sort of leading the country in terms of liver disease-related mortality increase, which is unfortunate. So a little bit about my background. I was trained as a structural biologist during my PhD time, and I went to med school afterwards. And I studied the lipids and the lipoproteins during my PhD, and I thought that I can apply this to fatty liver disease. And I started to study steatotic liver disease. As I move on into my clinical career, I witnessed really the surge of alcohol-related liver disease, which is another form of steatotic liver disease. So my lab studies both metabolic dysfunction-associated steatotic liver disease as well as alcohol-related liver disease, especially alcoholic hepatitis. So why are these two diseases important? So this is the landscape of liver transplant in the United States across over years. This is a more recent summary of the transplant data published in 2023. You can see that we used to live in an era of liver transplant dominated by hepatitis C, which is shown in gray here. On the left is liver transplant for liver cancer, which is about 20% of the liver transplant. And on the right is non-cancer end-stage liver disease liver transplant. This is about 80% of the transplant we do. We used to, a decade ago, we used to live in an era that's dominated by hepatitis C, for both non-HCC as well as HCC-driven liver transplant. But this has gradually been taken over by two diseases. One is alcohol-related liver disease, which is shown in green, and one is NASH, now we call metabolic dysfunction-associated steatotic liver hepatitis, NASH. So these are really the main driver for liver transplant and the liver disease-related mortality and morbidity. So during my clinical training, I started to learn how people study liver disease, and I joined the lab that does in vivo study using mouse. But I found the mouse model has a lot of problems. First of all, mouse model can mimic some human liver disease, but cannot recapitulate with high fidelity of the human liver disease state. It's pretty hard to get to the state of decompensated liver disease with portal hypertensions. Especially, it cannot reproduce. Right now, we don't have a mouse model that can reproduce alcoholic hepatitis, which is a subacute form of liver failure, which is a lot of the patients we see on wards. One of the projects I study is adenosine deaminase 2. That, unfortunately, is a protein that does not exist in mouse. It's a human gene that for primate only. And the last thing, probably also super important, is that a lot of the therapeutics in liver disease nowadays leverage GalNAc siRNA, which I will explain a little bit more. And the siRNA therapy oftentimes is a sequence specific. It's a species specific. So you cannot really test your agent in mouse. You have to use human. So this sort of a search for human model is echoed by a recent NIH as well as FDA push toward finding human-based research technologies. So this is an announcement made by NIH in April that they are trying to prioritize human-based research technology. And I think precision-cut liver slice very much fit into this category. It should be a technology that we should put a lot of effort on developing. So my lab used siRNA technology to find treatment for liver disease. A lot of you probably do that too. You know that siRNA can be transfected into cells using lipofectamine or any liposome-based technology. To do that, this is a technology we use in screening to identify the candidate siRNA, or in sort of a preclinical in vitro studies, in cell studies. But when it comes to in vivo, and when it comes to developing therapeutics, liver is really the prime organ. For RNA therapeutics, there has been half a dozen siRNA drug approved by FDA now. A large part is because of the GalNAc technology. So you can conjugate siRNA with a 3-sugar moiety called GalNAc, and the GalNAc has a liver-specific receptor, hepatocyte-specific receptor called ASGPR. And the GalNAc can guide your siRNA through the ASGPR and be delivered into hepatocytes very specifically. So this way, you don't have any contamination across other cell types. The drug is specifically delivered to liver cells. This allows you to treat a lot of the genetic problems, as well as getting into the mainstream for treating common liver disease like MASH. So we discovered a gene that we felt it's interesting and important for fatty liver, and we test that in host 7 cells, which is a human hepatoma cell line. And we found a couple of candidate siRNA, and it works very well in host 7 cells. But the problem is that there is not a platform that allows us to do the GalNAc drug delivery. Most of the hepatoma-based cell lines, including HepAG, we tested the GalNAc-mediated siRNA delivery just does not work. And we even tried to overexpress the ASGPR, and that does not do much. So one cell line does work is the primary human hepatocytes, but that is very expensive. It's batch specific. You have to find the right batch that will work for you, and it only works for the first couple hours. So you have to basically do the drug delivery very quickly after you plate the cells. And if it works, it works. If it doesn't work, then just have to try it again. It's very costly and not a lot of control in that experiment. So we start to look for alternative models. As you well know, that spheroids and organoids are very popular these days, and we tested the spheroids. The spheroids, these are spheroids by Inspera, and they use mature human liver cells to bioengineer spheroids, and we also use iPSC-derived spheroids that we make in our own group. And unfortunately, these cells are just not mature enough to have sufficient GalNAc-mediated drug uptake. Another approach is to use liver on a chip. It's another very popular human tissue model. But this is, to me, this is just a fancy co-culture system that with hemodynamics control. So whether you get a GalNAc delivery is really based on the maturity of the cells and how well you can sustain the maturity of the cells. We haven't tested it extensively on liver on a chip, and I cannot comment much on this platform. So about 2 years ago, I got in touch with Abby and explored the precision-cut liver slice, and then we bought a vibrotome and I started to do that, and there's no turning back afterwards. So this is leveraging my expertise as a hepatologist. I'm a transplant hepatologist, and I work at Beth, which is one of the leading liver transplant center in New England. So in the past 2 years, we've done the most liver transplant in New England, which is zone 1. And we rank nationally number 3 in terms of transplant outcome measured by 1-year survival. And the one unique aspect is that Beth Israel is the only New England academic donor service program, essentially OPO. So we do donor organ procurement locally in our hospital. We have a donor service program that we take care of a donor patient as well as donor family to procure the liver organ, and then we distribute the liver organ to other transplant centers. So I have established a end-stage liver disease registry to enroll patients that allow us to get access to liver tissue. So this registry basically enrolls patients with all form of liver disease that is end up waiting for liver transplant. So our center do all kinds of liver disease related to liver transplant, including MASH, alcohol, viral hepatitis, autoimmune-based liver disease, autoimmune hepatitis, PBC, PSC, drug-induced, acetaminophen-induced liver failure, as well as sometimes rare liver disease like polycystic liver, a situation like that. So we have access to a lot of the recipient explant liver and sometimes donor liver as well. So for the recipient liver, we basically have the entire liver at our disposal, and we can get as much liver tissue as we want. And for the donor liver, our transplant surgeon has been gracious enough to offer the caudate lobe using a University of Toronto protocol that they take out the caudate lobe, but they don't transplant caudate lobe. Caudate lobe has a different blood supply that's not directly supplied by the portal vein. So taking out a caudate lobe does not really impact the quality of the outcome, but certainly it's one step additional in the transplant process and potentially can increase bleeding risk. So something you have to be careful about. So we get liver tissue from the OR, and then we bring to the lab and slice the tissue. So this is something that you probably do in your own lab as well. So the liver tissue is punctured into a cylinder form. We use a skin biopsy device to get a puncture of the liver tissue in the cylinder. This is usually about a 7 or 8 millimeter diameter tissue. And then this is embedded in agarose and very rapidly cooled. And then we put on the vibrotome. We have the VF510 ultra zeroing vibrotome. It's also known as a compressotome. So the device actually gives pressure when you slice it. So this is a little bit better than the Leica vibrotome device. So we transport our tissue in University of Washington solution. So this is a specific buffer we use to transport liver organ for transplant. We can preserve the organ in cold UW solution for many hours. Back in the old time, we don't have the liver perfusion technology. This is the prime way of preserving liver organ. So all the transportation of liver tissue, as well as the slicing, are done in the UW buffer. After slicing, you get a tissue like this, and then we put them into the 24-well format with tissue in each well. And usually in one batch, we can get about 4 to 5 plates of PCLS liver tissue. Any question here? This is, I know this is the technical part of things, and probably it's of interest to a lot of people.
Hey, Gordon, I had a quick question. I just wanted to, since you're talking about live slices, and also you talked about spheroids and liver on a chip, can you tell us a little bit about the viability and expectation for these different types of like tissue models, tissue models?
Yeah, so don't worry, I will get in depth about the viability and what the tissue is doing in culture. Yes.
Fantastic. Thank you.
This is what this webinar is primarily about. So when we first start doing this, obviously we're interested in the viability, and we don't have a good way of measuring viability. We initially rely on morphology, and we look at the literature, and it seems like a lot of people, most literature, are only using the tissue in the first 3 days. Some extend to 5 days. Very few people extend beyond 7 days. So what we have observed is that as you culture the tissue, over time, the tissue will collapse. The tissue will sort of aggregate. And just without much of scientific basis, to me, this is a sign that the tissue is not doing well when they collapse. So we start to optimize, and we sort of optimize the protocol. Now we can routinely get tissue to culture beyond 14 days with a morphology that is maintained. So what are the parameters that we think is important? I think the most important thing is ischemia time. The healthy tissue you start with, the better outcome you get. When a tissue has long ischemia time, especially the warm ischemia time, the outcome is bad. The tissue tend to sort of aggregate early. Tissue preservation during cutting is very important. Again, the temperature is very crucial. Cold ischemia is a lot safer than warm ischemia, so you want to keep the tissue cold. So all our cutting is done on a cold plate. All our pre sort of slicing cutting, preparation the tissue is on a pre-chilled metal plate, and all the buffer that we use has mixed sort of the we sort of freeze the UW buffer, and we has the ice slurry, and we mix the ice with the buffer, so temperature is always near zero when we cut, and then the tissue is always preserved in cold buffer. So usually from the time that we get a explant liver or a caudate lobe outside of the warm body to a PCLS tissue in culture, usually the time is less than 2 hours in our hands. We bring the tissue back, we process right away. So this, to me, is the most important thing to give you good outcome. So we implemented a post-cutting tissue recovery. So this is step that you use a hyperoxygen. We use 95% oxygen for 3 hours to promote tissue recovery. This is a trick we learned from the literature. We also optimize the culture condition. We have special media to culture this liver tissue. Right now, we use 20% oxygen in culture, and they are cultured in a shaking modality, as suggested in the literature. We are testing different oxygen concentration. We have procured a tri-gas incubator that we can control the oxygen concentration during long-term culture. And we hopefully will find optimal oxygen supply that will help the tissue. And in terms of the optimal dimension of the tissue and how much buffer you should use and the thickness, this has been well described in the literature. So usually the tissue is no bigger than 1 centimeter in diameter. And the optimal thickness we find is about 250 micron. And the media gets very expensive. So we found that 1 ml is better than 500 microliter. It adds to the cost to the overall production, and it's especially if you want to culture for a long period of time. So viability, is the tissue live? So conventionally, if you look at the literature, people analyze viability by ATP assay. We did ATP assay as well. I really hate it because it basically wastes all your PCLS. You need a few, n equals maybe 5 to get a reasonable standard deviation. And then you need to do it at multiple time points. And you basically waste all your tissues just to know whether they are alive or not. And if there's no gold standard cutoff, you have to establish your own cutoff to understand what ATP level is considered to be good, right? So we developed a new method of measuring viability just by taking one tissue at any time point you wanted to know the viability. And we do a whole mount live tissue staining. So this is the live tissue that we stain the tissue live with two dye. One is calcium AM. So this is a dye that's freely permeable to the membrane. It will get into all cells, but it requires a esterase. The cell has to be live to produce the esterase to convert it into a green fluorescent color. So the cell has to be live to make this enzyme to give you the color. So if the cell stains green, the cell is live. And this green has a variability in intensity. If the cell makes a lot of the esterase, it will be brighter, basically. And you also have another dye that's acidium homodimer. So this is a dye that's not permeable to the membrane unless the membrane leaks. This dye stains the DNA. So if the cell is dead with holes on the membrane, this dye will get in and will stain the cell red. So this is a PCLS from a healthy liver tissue. You can see that most of the cells are live on day 3. So this is using confocal imaging for the whole tissue. It's a very big tissue that we imaged. In this particular PCLS, most of the dead cells are on the periphery. So you can see there are dead cells embedded here. And whenever you have a sort of an irregular piece that's hanging off, that piece tends to have a higher amount of dead cells. And I wonder why that is the case. I think this is probably coming from the preparation of the tissue in a cylinder format, because we use the skin biopsy device to get a piece of liver tissue. So there's going to be some crushing injury. I think this is probably mostly coming from that crushing injury. And this is something that I tell Abby in the past as well, that if we can engineer a device that will give you a cleaner cut, this will help with the quality of the tissue. So
Thanks, Gordon.
So this is the time series that we do. Please remember that it's not the same PCLS. Every time you do this, you're going to take a fresh PCLS. And so there may be some variability from tissue to tissue, but it will give you a good sense of what you're looking at. So it starts with a very high quality tissue, but over time, in day 3, in day 7, you start to see some patterns of the cells that are starting to die. So here you can see streaks of dying cells, right? And in day 10, you can see sort of lobules that are dead, right? And then in day 14, in this particular tissue, the periphery are mostly dead, but somehow the cells in the middle are still alive. So even in day 14, you still have a lot of live cells when the condition is right, when you prepare the PCLS right. So that is very encouraging. So if there's one message you're going to take away from this, is that a PCLS is an injury model. It's not cell culture. The cells are changing over time. So it is important to understand what you're dealing with if you're going to use PCLS in your experiment. So we find active things happening in PCLS. So if you look at the literature of people working on PCLS, they will tell you that the tissue gets more fibrotic over time. And we saw that in healthy liver tissue-derived PCLS. So let me walk you through some of the changes we see. So this is close to freshly cut. This is day 1. And you can see that the tissue just looks like a liver biopsy. You have very beautiful liver parenchyma. So this is a portal area. You have the bile duct. You have some cells in the bile duct. You have portal vein. If I look at this, I will not be able to say this is PCLS and this is not a liver biopsy. It looks very healthy. In day 3, in the first couple of days, the first thing you notice is that there are gaps between cells start to form. There are white lines between cells, right? I'm going to get into what they are. If you look at day 7, interestingly, the bile duct is still beautiful. It's intact. If you can find a bile duct in the tissue, usually they still look intact. So the bile duct morphology, despite there's no flow in the bile, they're fine. The portal veins, the structure is largely maintained, hepatic arteries. But you can see that the portal area is getting bigger. This particular portal area is much bigger than what you anticipate initially, indicating there's probably some liver fibrosis. And if you look at a lot of tissues, you will very easily find on day 14 this very thick fibrotic band. This almost looks like a cirrhotic tissue. So we also done series red staining, which stains for the fibrotic tissue, and I didn't show it here. The fibrosis is portal fibrosis based. This is not it's not a predominantly sinusoidal fibrosis that we see. It probably has to do with this is a live tissue, but it's a little bit of constraint. You don't have the full mobility of cells. The cells are sort of stuck in where they are. You don't have infiltrating immune cells. You're basically stuck with whatever immune cells you have in the tissue already. So these cells are predominantly in the portal area. The hepatic static cells are predominantly at the portal area. So you end up getting a more portal fibrosis-driven pattern. You do have some sinusoidal fibrosis, but it's milder compared to alcohol and the NASH disease that when you see a lot of sinusoidal fibrosis. But fibrosis in a healthy liver tissue happens over this time period, and you will see that around day 7, and it gets pretty extensive around day 14. So, so this is using a special stain called the reticulin stain. We use this a lot in clinical pathology in liver disease, but this is not used often in liver research. But this is a very useful stain. This is a stain that stains type 3 collagen. It allows you to see the overall structure, the tissue architecture very well. So liver is not a random sponge. The cells are organized in very highly structured way. The liver cells, as you know, are polar. Their apical side is facing the canaliculi. In between cells, there are tight junctions. The cells are not, the stuff are not leaking through the tight junction. On the basolateral side, they face the sinusoid. That's the circulation. So usually, in a lobule, the liver cells are organized in plates. The plates has 2 layers of liver cells with the canaliculi in the middle, and then they sort of coalesce into the bile duct. And then you have the sinusoid. So when you stain type 3 collagen, stain the basement membrane, you get to see these plates. You can see sort of the double cell thickness of the liver cell plate, hepatocyte plates. And this is sort of it's not parallel. It's sort of being a very complex 3-dimensional structure. So you don't get a clean cuts. You get this sort of a network. In this particular case, you can see this portal area. You have the bile duct very beautifully done, and you don't have the basement membrane between the cells. It's on the basolateral side, and then you can see this hepatic arteries, and they have this central vein area, and you can see the sinusoid. The space sort of goes into the central vein. Now, if we do this on the liver tissue over time, you can see what is happening. So initially, in the first couple of days, as I told you on H&E, that you see there are gap between cells, and this tells you what is happening. So first of all, the basement membrane just does not disappear. Whatever basement membrane type 3 collagen is there, it's always there. It's there across the entire culture. So these things does not get degraded. So you can see that the space between the hepatocyte and the basement membrane are being created over time. This sort of makes sense because the nutrient is no longer flowing through the sinusoid. The nutrient is basically everywhere. The cells are soaked in nutrient. I think in PCLS, the polarity of the cell probably is altered in the first couple of days right away because the nutrient is coming from everywhere. So there's no need for the cell to adhere seamlessly to the basement membrane. It's to the advantage of the cells to create a space to allow nutrient to come in all directions. So you see that there are space created. These are not only space between cells, but these are also space between cells and their basement membrane. In day 7, you can see that at the basement membrane staining, some area getting thicker. In the portal area, there's a lot more collagen staining here. But very interestingly, in day 7, sometime around day 7, you start to see focus of cluster of cells without basement membrane. So these are cells, are new cells, in my opinion. These are not cells that we brought from the original liver state. So liver regeneration is happening in PCLS. So this is a model that you have active liver regeneration. And it creates this area of liver cells that are new liver cells from division. On day 14, in a healthy liver PCLS, you have a lot of these areas. This is super interesting. You're dealing with a model that is live. Liver cells actually growing in PCLS. Any questions so far? All right. So one advantage of the PCLS is that you have a lot of cell types. So you can stain for different cell markers like CD31, CD68. CD31 is for endothelial cells. CD68 is for macrophages. Liver is the largest innate immune system organ. 80% of the macrophages are in the liver in the form of Kupffer cells. There's a lot of Kupffer cells, and you can see that you get the intact blood vessels, sinusoids, as well as the blood vessels shown here. This is probably artery, and this is probably a vein. This is probably also vein, right? And then you can see clusters of Kupffer cells shown in yellow here. This is probably a portal vein. These are small nuclei without stain. These are probably lymphocytes. So sometimes you can see the immune foci. This is a picture that I thought was quite interesting, that you almost got a shape of a vessel here, stained by CD31, and then you have a cluster of immune infiltrate with infiltrative monocytes and derived macrophages. Yes, in clinical pathology, we call this portal macrophages. These are probably lymphocytes cluster. There's an immune infiltrative immune focus that you can find in PCLS. These things will exist for days. I don't know how long exactly they exist, but from day 3, day 5, you can easily find these things in the tissue. So Abby mentioned seahorse. Seahorse is the ultimate way to measure viability. For those are not very familiar with seahorse, so this is a technology that allows you to directly measure oxygen consumption rate. You need to have live tissue to consume oxygen through the mitochondria, and the seahorse will be able to measure, number 1, oxygen consumption, and number 2, the pH change. Right? So seahorse is primarily designed for doing cell culture studies. Need a very even confluent cell to do reliable measurement. It's hard to do that on a tissue. We used a islet capture microplate. So this is a plate that's specifically designed for pancreatic islets, which is also a tissue. And they have this mesh device that allows you to lock the tissue under at the bottom of the plate. So this is a lever you use to apply this mesh plug. So you put tissue in the bottom, and then you put a plug on top of that. Fortunately, the size of the PCLS sits pretty nicely. It's a little bit bigger than the well, but I would say 80-90% a good fit in the bottom, and then the mesh can cover that. In a seahorse experiment, you use inhibitors of different complex of mitochondria. So this allows you to tease out different compartment of mitochondrial activity. We use oligomycin to measure the ATP-linked oxygenation, and then you use FCCP to get the maximal oxygen consumption. And then you use the ontamycin protein to sort of inhibit everything. So this is what you get for a day 6 PCLS, and then you get this beautiful response to the inhibitors of mitochondria, indicating that a lot of this activity is coming from mitochondria. Notably that this line does not go all the way to the bottom. you usually see in a cell culture. And we were initially worried about this. But as we look at other sort of data from tissue, this is actually very common for tissue because it's very hard to get inhibitors to go all the way into the tissue in a short duration of the experiment. So in the tissue, you use much higher concentration of the inhibitor, and they may still not 100% penetrate into the tissue. This is something that I think we need to work with Agilent to see how do we come up with the optimized protocol and the standardized reporting, and what is to be expected as the best practice, and what is expected to be the readout you get. I think this is working. Regardless, you can just measure oxygen consumption rate. You don't have to bother with all these inhibitors. This tells you how active is the tissue, and you normalize by the total amount of protein, and you can see that the tissue is a highly metabolically active and consuming oxygen. So this value of 400 is pretty high compared to what you usually get for cell-based seahorse experiments. So in this particular experiment, the maximum we got is among these three data points: day 3, day 6, day 9. Day 6 has the highest oxygen consumption rate. Day 9 is the lowest, probably indicating that some cells are dying, despite some new cells are being made. So what I have talked about is healthy liver tissue. But I think a unique opportunity for PCLS allows us to directly access diseased liver. So you don't have to sort of find an animal model of disease. You can directly work on diseased human liver. And we have all kinds of liver disease for you to study. So I think looking at a diseased human liver is super, super helpful. So if we just look at the overall morphology of healthy versus diseased liver, healthy liver PCLS is very homogeneous. It had this brown sort of color over that. It's because the portal area is relatively small. It's not a lot of fibrosis, and it's just evenly, hepatocytes. More than 50% are hepatocytes. In a diseased liver, when we say disease, oftentimes they have cirrhosis. There's a lot of scar. The scar is going to be a lot more light translucent. So this light area, the scar area, these are the micronodule. So these are the liver parenchyma. The brown part is the liver parenchyma, and the white part are the sort of the fibrotic area. So morphologically, they look very different. So how do they look over time? So we developed a way that you can image just the gross picture of a slide over time. So we can take picture every day, and then you can see how the morphology change. So this is from a healthy liver. You can see how it's changing. So you can see that it starts with a very homogeneous brown tissue. To day 14, you have area that's pretty white. This is probably a lot of fibrosis. And then you have area that's very, very brown. There's probably still live cells, and probably some regeneration happening. Right? I can play this again. Right? So what about diseased liver? So this diseased liver, you can see that has a lot of micronodules. You can see how this is doing. So for this tissue, we went all the way to 21 days. Not a lot of changes, right? The white area is still white, and the brown area is still brown. The change is not obvious compared to the healthy tissue. So this is exactly what I see on histology. So if I cut these tissues at different time points, so this will be different tissue, obviously. So it's not directly comparable. It's not the same tissue that you look at over time. Because it's a disease tissue, you have a lot of fibrosis to begin with. Some inflammatory cells you can see. You have that all the way across. And I've been told by some people who have worked on PCLS, and they tell me that if you culture it more than 7 days, you just end up getting a piece of scar. And that's not what I see. For disease tissue, the fibrosis does not take over the entire liver. The lobule is pretty well maintained. It's pretty static, seems like. So this is the case of a tissue that has fatty liver in it. I had to wonder what happens to the steatosis. The steatosis go away because the metabolic environment is totally different. It turns out that at least for this particular tissue, the steatosis does not go away. The steatosis is persistent. It's persistent over the entire duration of its culture. We have all the way to day 14, and you can see steatosis all the way there. So this indicates that in a disease tissue, a lot of the disease features are actually captured. They are probably maintained in a big level over the duration of the culture. So this is doing our true dye staining of a diseased liver. You can see that at day 3, day 7, it look pretty good. Day 10, you start to get some island of cell death. These are some lobules. Day 14, it gets a lot more. And then day 21, it's pretty much most of the tissue is dead. So this is probably the end state you get when most of the lobules have dead cells. But you probably still have 10 days of cells that are pretty useful. Right. So we can also do the seahorse experiment, and you can measure the oxygen consumption in diseased liver tissue. If you look at the scales, the diseased liver does have a lower, despite it looks bigger, but this is actually a lower number compared to healthy. It's lower oxygen consumption compared to healthy tissue. In this particular disease tissue, we did not see a decline of oxygen consumption. In day 9, it's arguably a little bit higher, probably due to some regeneration of liver cells. Right. So, this is a exciting journey, and we are learning PCLS as we keep trying it. We've done 20 batches of PCLS so far, and every time we learn something new about this technology and how the tissue is doing in culture. But the most exciting things is go back to the original reason that we went for PCLS is GalNAc-guided drug delivery. This is probably by far the best model to study GalNAc-guided siRNA delivery. So here we use a fluorescent labeled GalNAc. This is site 3 label, and then we give this GalNAc to I think this is a day 3 diseased liver PCLS. You can see the nodules of the liver. You can see beautiful uptake of the drug. You don't see that in any cells that we studied or in organoids that we studied. It's a beautiful drug uptake. This reflects the amount of receptor expression in these cells. And if you look carefully, the uptake of GalNAc siRNA actually follows isolation pattern. The uptake is higher in zone 1, that's the periportal area, and it gets weaker in zone 3, which is the pericentral area in the middle of the lobule. You can see this gradient more clearly in the zoomed-in views. Right. So the drug is actively being taken up. The gradient probably reflects the phenotype of the hepatocytes. The zonation phenotype of the hepatocytes probably are maintained across several days during the culture, that you have more receptor on the hepatocytes in zone 1. So back to our experiment, we saw beautifully the GalNAc siRNA that we identified successfully suppressed the gene expression in PCLS, which we cannot accomplish in previous experiments. So I think this is a very good model to study this type of things. So what's the conclusion? The conclusion is that we have refined a PCLS protocol that now can culture PCLS for a long duration of time. You're not limited for the first 72 hours anymore. You can plan a much complicated experiment in that setting. This also gives you opportunity to potentially build models using PCLS. So we find sort of slightly different behavior for PCLS from healthy donor versus from cirrhotic liver. For PCLS from healthy donor, there seems to be a spontaneous liver fibrosis happening. So my interpretation is that this is a native liver response to injury. PCLS is probably the biggest liver injury you can expect on a liver. You basically are chopping liver into pieces. So the liver has to respond to it, right? You're changing the cultural environment. A healthy liver will have a way to respond to it. You have the immune cells, Kupffer cells that are not exhausted. You have the hepatic stellate cells that are quiescent that can be activated. So a lot of this machinery in response to injury are intact. And what you see is fibrosis, which is a natural response to injury. And what you see is liver regeneration, right? In cirrhotic liver, there seems to be a lot less of that, probably because this machinery are already exhausted. You have the disease state of what you have. This is an advantage and a disadvantage. Advantage is that you have the disease feature that is already captured in the disease state, and the disease probably will carry on for a long period of time. But the disadvantage is that you don't have much of these dynamic changes, which you may not want in experiment, right? I think this creates opportunity for you to build a model for, like, say, hepatitis. You can combine this with alcohol and see how the healthy liver reacts to alcohol as well as this injury. And you will get a spontaneous fibrosis, which is something that people try very hard to recreate in mouse. But you don't need to do anything else. The liver will spontaneously fibrose over time. So we're studying antifibrotic drugs. This is perfect system to study antifibrosis. So I think this is a very versatile experimental platform for you to customize it to your need to study your biology and test your drug. So what are we doing in the lab in the future? I think still a lot need to be learned about what these tissues are doing in culture. So we are working with the vendor of the media directly, and we collaborate with them to see whether we can find a way to come up with a media that will provide even better support. Right now, we use a media that are predominantly optimized for hepatocytes. So it will give maximum nutrient support to hepatocytes and helps hepatocytes to grow as long as possible. But it probably are not sort of optimized for other cell types. So there are probably room to improve the media support for other cell types. This is a big challenge. PCLS is a big challenge for biliary epithelial cells and eosinophil cells, because you don't have the tubular structure anymore. So the bile flow is not there, blood flow is not there. How these cells react to this new environment is something that I think will be of interest and should be studied. As I told you that I'm studying the impact of oxygen on PCLS culture, and we're playing with different oxygen quality of the tissue. But I think ultimately we should study the dynamic changes of different cellular landscapes in PCLS over time. So this requires a little bit of investment to study this using a single cell RNA sequencing and spatial transcriptomics, spatial proteomics. PCLS is a great platform to use spatial technology. You can capture this sort of the spatial relationship of cells in PCLS. So these are the things that we are doing in the lab to report to you in the future. So this is really a teamwork. It takes a lot of effort. We have a vibrant team with a lot of young scientists, trainees, very excited about technology and innovating. And I want to specifically mention Sophia. Sophia is the person that started this project and did a lot of optimization. Very quickly learned this technology from Abby and adopted a lot of refinement in the protocol. Other people have helped to develop different analysis modality of PCLS. So this is a very close collaboration with our transplant team. This is our transplant team, and they are the ones make this transplant program really superb. And most importantly, the dedication is to our transplant patients. I have yet to meet a transplant patient that is saying no to me for doing this, for taking their liver. It's their generosity to use their organ, make this project possible. And I will stop here. Thank you.
Thank you so much, Dr. Jiang. And I also wanted to thank Sophia as well. I worked with her and communicated with her for years now. It's been a pleasure supporting your lab and vice versa. So we do have a question from Dr. Jay Kim. And Jay, would you like to unmute yourself and ask your question? Please feel free to.
Okay, my question is, what kind of a medium do you use to make a PCLS viable for 14 days?
Yeah, so we use a media from Takeda. I think it's called a Ceratis. Yeah, this is a hepatocyte media. Let me find its full name. And we are actively working with them to develop a more customized media for PCLS as we speak.
What about the media that is used to transport liver organs, for transplant? That's for human livers rather than, say, a murine model. Have you found incubating or using that type of media, which I know is very expensive? Is there an advantage at all to that?
So there's a reason University of Washington media is developed for liver organ transplantation. So this media, it's really a buffer. It's not a media. It does not have nutrient in it. So this buffer has ATPs, has glucose, sort of maintains the basal sort of the support of survival of the cells. So I think this buffer is very important for preserving the cells when they don't have media support. And I just pasted the media that I use for cultural PCLS there.
Thank you. I think there were some more interested questions, perhaps. Anybody else? I don't know if somebody from the Schmid lab had shown their video and was interested in asking a question.
Yes.
Hello, yes, I have a question. Hi. I also work on tissue slices. I've been doing tissue slices from liver and also primary tumor. So I was wondering if you just place the slices in the well or you use some kind of support, because in the past I was using some cellulose membrane, and then most recently I've been using insert, because I've noticed that when the slices sink into the well, then they are not anymore exposed to the oxygen, and then the necrosis occur in the samples. I don't know if you had this issue as well, or you managed to avoid it in any other way.
Yeah, so we use the way that Abby taught us. We don't use any additional support. We try to leave the agarose there with the tissue. So the agarose will serve as a support. So the tissue is not going to bounce against the wall of the well. And it basically floats in the media. I don't think it sinks to the bottom. It just floats there. And that probably gives you more oxygen exposure. And it's cultured in a shaking mode.
Yeah, the shaking is what helps it kind of stay suspended. I think one problem was if you have depends on how large diameter of a core you're taking. It's sometimes the tissue can fold onto itself as it's shaking. So if you leave the agarose rim from the compressed system after you section it, it prevents kind of that folding over. Okay.
Yeah, no, thank you. Probably our slices are much bigger, because even if I leave the agarose, then my slice.
Yeah, it depends. And then you can use like the 12 well plates or the 6 well plates, maybe move up to something a little bit larger to suspend them in. Yeah.
Okay, thank you very much.
Oh, thank you. I just wanted to reach out and ask if there are any other questions or comments for Professor Jiang's talk today. And if not, please feel free to reach out to me and etc. And so
William Jawa had a question.
Oh, great. Let me take a look. Oh, I don't see it in the regular, but go
Hello.
ahead and
Hi, thank you very much for the nice presentation. You presented or you showed that cells are viable for 14 days. It was very nice shown. But what about the functionalities? Did you check for liver-specific functions or the cytochrome P enzyme's activity? Or
Yeah, I didn't show the data there. We are looking at some of the functions, like for instance, we measure the albumin production. So a lot of things happen in the first couple of days. I think the albumin production, for instance, would decline initially, and then they stabilize, and then may slowly go up, and then it completely drops in the end. So there is a dynamic happening. I think initially the cells gets a shock, so the function probably are impaired a little bit, but then the cell gets used to it, and then you have the regeneration kicking in, sort of helps. And eventually everything dies out. So you're basically dealing with this dynamics. But I have not checked every sort of. Liver have so many functions, and I have not looked at all these functions.
thank you.
thank you. So I think I will wrap it up unless there are any more burning questions. And I just want to say a huge thank you to you, Professor Gordon Jiang. And I think we'll stay in close touch, and maybe we'll come back and do an update for new things, ways for updates for PCLS. Great.
Anytime.
Take care, everybody.
Thank you for having me here.
Of course.